So today's session is looking at different topics around forensic biology and DNA, which means I am at the correct podium because I am the program manager for NIJ, working with all of the grantees around forensic biology and DNA. So your first talk today is by Dr. Ashley Hall and Dr. Ray Wickenheiser. Dr. Hall is the director of the Forensic Science Graduate Program at the University of California, Davis, and Dr. Ray Wickenheiser is the retired former director of the New York State Police Crime Lab System and is now the principal consultant for the Ray Wickenheiser Forensic Consulting.
Thank you. And hello. We are here today to talk to you about trace DNA and activity level propositions and introduce you to our bullpen of events. All right, disclaimer, the views that we express are our own. They are not the American Academy or either one of our institutions, present or former. All right, so let's talk about activity level reporting. In evaluative reporting, the forensic scientist focuses on the probabilities of observing evidence given alternate scenarios or propositions. They need to draw on their training, education, experience, and empirical data to aid the trier of fact in their deliberations. What we are specifically going to talk about is empirical data. There is a real lack of empirical data out there in the published literature to support evaluative reporting. So that's why we've developed our system to generate this data for the analyst. So we're working specifically with trace DNA. Now, the problem with working with wild fingerprints, that is fingerprints that are naturally deposited, is that they are so variable. We don't know how much DNA they contain. We don't even know where the DNA actually comes from. They vary wildly between people, and within a person, the same person can deposit wildly different amounts at different times of day. So we can't do any real quantitative work with natural fingerprints. So we have developed the domesticated fingerprints and the domesticated hand as a transfer vector. We use the data that we generate with our domesticated hand and fingerprints to populate the bullpen of events, which is a repository for empirical DNA recovery data from trace DNA samples in real-life activity scenarios. This is the data that can be used to support the forensic scientist in their comparison of alternate activity level scenarios. All right, the domesticated fingerprint is a ground truth sample. There is a known quantity of DNA contained in a background of sebaceous fingerprint chemistry. Using the domesticated fingerprint, we look at the three key fail points during collection and analysis. That is, DNA left on the surface, DNA left on the swab, the collection device, or DNA lost during the manipulations of extraction. Every time we do this, we lose DNA at every point. We've been able to quantify how much DNA is lost with a particular device or a particular extraction technique. This allows us to better track the DNA in the domesticated fingerprint across a transfer pathway. If we account for all of it, we can have more confidence in our results when we're speaking to the actual transfer and recovery activities. This has been published. This is where you can find it to read more. Exceptionally well-written paper, I gotta say.
Unbiased, of course.
And this is what our bullpen looks like. Now, this is just the beginning of it. We have a long way to go, but you can see that we have different surfaces. The different skin is what we put on our domesticated hand. Key fail points, like we lose 25% of the sample to a standard cotton swab. We lose 49% during a DNA IQ extraction. So a lot of the DNA we have shown is lost just during even excellent collection and analysis. So we've got the domesticated fingerprint. We've got a way to control the amount of DNA that's going into it. Now we need a way to actually affect transfers. If we use a natural hand, a wild hand, the problem is that it brings its own DNA to the table. It brings its own DNA and it would dilute the results. So the domesticated hand, we looked through the literature. There is a lot of mock skin out there. We chose 3 formulations that are closest to the frictional activity of skin, of true skin, because that's one of the most important factors in transfer. We experimented with the 3 and narrowed it down to this, the Lorica leather. Lorica leather is attached to a nitrile glove with Gorilla Glue, and they're used as the mock hand. We have taken to only adding the Lorica leather to the parts of the hand that are involved in the transfer. So in a handshake, we have to have it on the fingertips of one hand and then here on the other hand so that we can do the transfer and follow it through. We call them the domesticated hand, the dom hand, and the mock hand, the mo hand. That is just to differentiate them. We typically put male DNA on the dom hand and female DNA on the mo hand, right? So later on, we can do quant trio and we can separate out the components. So the goal of the domesticated hand, the domesticated fingerprint, is to look at DNA transfer pathways, look at how primary, secondary, tertiary transfers look compared to each other by quantifying the DNA recovery. This is the empirical data that the DNA analysts can use to inform activity level propositions. So using these two mocks, we've taken out the human factor, right? We've taken out the unknown quantity of DNA, and we've taken out DNA that gets added during the transfer. And now we use our mock hand and our domesticated fingerprint to mock up true crime scenes. These are realistic scenario activity studies to generate empirical data in real-life situations. And we started with the case of Idaho versus Brian Kohberger. So this is the case where 4 University of Idaho students were murdered in their off-campus residence. They were stabbed. A knife sheath was found at the crime scene, and Kohberger's DNA was found on the snap. So one proposition could be Kohberger touched the knife sheath snap. This is a direct transfer. A second proposition could be Kohberger shook hands with another person who touched the knife sheath snap. This would be an indirect transfer. Now, this is not the only indirect transfer that would be possible, right? But it's the simplest one, and it's the one that would transfer the most DNA. It's the most direct, indirect transfer we can do. Therefore, it should be the most conservative, which is what we're looking for, a conservative estimate. So this is what it looked like. We placed a domesticated fingerprint on the domesticated hand. The domesticated hand then touched the knife sheath snap in a direct transfer. For the indirect pathway, the dom hand shook hands with the mo hand that then touched the knife sheath snap. And so remember, on the dom hand is male DNA, on the mo hand is female DNA. We collected samples from all of the surfaces so we could track the DNA through the transfer pathway. With wild fingerprints, a male hand touched the snap. For the secondary, a male hand shook a female hand. Female hand then touched the snap. Now, pulling it all together, I've got a reminder of what the pathways are. For the domesticated fingerprint, comparing the direct versus the indirect, the mean male DNA recovered after the indirect pathway was only 44% of the male DNA recovered after the direct pathway. All these numbers that I'm showing you are important because we followed the DNA through the pathway, and I'm going to show you how we figure out how much of it in just a minute. But you can see about the same quantity of DNA is transferred, regardless of how many contributors there are. But in the second indirect, that ratio changes. The proportion of the male DNA that we're looking for is lowered by the contribution of the other DNA. With the wild fingerprint, we saw that the male DNA from the SNP after the indirect pathway was only 10% of the male DNA on the SNP after the direct pathway. So it was a conservative estimate. The domesticated fingerprint is a conservative estimate of what happens with wild fingerprints, which is what we want it to be. And we're seeing transfer of about the same amount of DNA, but a reduction of the male contribution through the contribution of the female, who is the final contributor. How do we pull it all together, and why is it important that we use the domesticated fingerprints? Well, if you see here to the side of the screen is the bullpen. This is DNA retention, so that is how much DNA is lost. You can put in the amounts that were recovered from each surface, and then the spreadsheet calculates for you how much DNA you're accounting for. In this case, it was 99.8. By factoring in, say, 25% loss at the swab, we were able to account for almost all of the DNA we put in, which gives us more confidence and the results that we're getting when we compare the indirect versus the direct pathway. All right, so this data goes into the bullpen of events. How do we use the bullpen of events? Ultimately, it would be nice if we didn't have to run the pathways for every different crime that occurred. If we could have a repository where that empirical data already existed. So let's say we're using the bullpen of events to look at event 1, event 2, and event 3. Now, these are independently occurring events, so we can multiply them. Because we have each one defined the loss that's due to the surface, the swab, and the extraction, they become independent of each other. So event 1 is, say there's a nanogram of DNA on the pole. Event 1 is a transfer from the hand to the pole. We know that there's an 85% of the DNA transfers. So that would be 0.85 nanograms transferred to the hand. However, when we use the bullpen data to calculate how much was lost, we see that we only actually get 0.22 nanograms. That is an actual recovery with the concentration of 0.0073 nanograms per microliter. But that works out to 0.11 nanograms we could possibly add in the 15 microliters that go into the AMP reaction. So the question to the analyst is, what profile do you expect to get from 0.11 nanograms? Expect to get a good profile, a partial profile. But this gives the analyst data to make those predictions. Now add event 2, that is hand to drug baggie. There's a 93% transfer. That would be 0.21 nanograms, but calculate the loss, and we get an actual recovery of 0.05 nanograms, or 0.026 nanograms into the profiling reaction. What sort of result do you expect to get from that? Now add a third event, transfer from a drug baggie to a second person's hand, and we get a recovery in theory of 0.049 nanograms, in actuality of 0.00043 nanograms, and add 0.0065 nanograms to the profiling reaction. So by combining the different scenarios that are in the bullpen, you can string together any series of events and do this kind of calculation, get the kind of data to do your predictions about what you expect. Now that's one way to look at it, is by quantification data. Another way to look at it is, what does the profile tell me? So we did 10 reps of each pathway, direct and indirect, and we looked at the sample that we collected from the knife sheath snap using PowerPlex. This is a domesticated fingerprint DNA profile, the result of a direct transfer. We see a major male DNA profile in 10 of 10 snap samples. This is the whole profile, and then a close-up of what it looks like. In the indirect transfer, you see a little bit of a different situation. We see a single donor female profile in 8 of 10 samples, and a detectable minor male profile in 2 samples at 3 loci, and this is an example of what it looks like. The arrows are pointing to the male alleles. All right, so direct and indirect transfers look very different, both by quantification and by profiling. Now for completeness, let's look at the wild fingerprints. We have a major male DNA profile in 10 of 10 samples from the knife sheath snap. In the indirect transfer, we get this sort of profile. A major female DNA profile in 5 of 10 snap samples. The other 5 samples included a partial male minor profile. This is what it looked like. There were 1 to 2 male alleles at 1, 3, or 6 loci in those samples. And the arrows are again pointing to the alleles that are attributed to the male. So what we see is the domesticated fingerprint is a conservative surrogate for the wild fingerprint. That data can be qualified and given greater, we can take greater confidence in it because we are able to follow the DNA through the pathway, and we're able to account for all of the DNA that we deposit. Between direct and indirect pathways, we see a marked difference in quantification values, and we could use those quantification values to predict whether or not we would expect a profile and the quality of that profile. And when we look at profiles, we see a difference in the direct versus indirect pathways. And thus, I could say, in this case, A major male DNA profile is more likely if a direct transfer occurred.
Okay, thank you, Ashley. So now I'm going to talk about human factors and the philosophy of trace DNA. So first of all, scientists, we are qualified to give opinion evidence. Note that the juries, if the defendant chooses to go with the jury, they could choose to go judge alone, but the finder of fact is typically, in the United States, the jury. So when forensic experts are qualified to give opinion evidence, that opinion via Rule 702 is that it's based on your education, training, experience. So we're probably familiar with going to the doctor. If you need potentially, you have a heart ailment, they're going to base that opinion based on data. You might get a slightly differing opinion depending on the doctor. You're still going to listen to that opinion, but it should be qualified as to why. Why do you need the heart surgery? So providing this data is the critical component. So you should express your opinion, but you should be able to say why, and that why is based on data, and we're thrilled that we're able to be supported through NIJ to provide that why to you. So the challenges become less about whose DNA is it, as to more why did it occur there. So we've all heard about activity level propositions. These alternatives are being given. We have placed the DNA there. It's not a proposition. This is data, so we're providing that information to assist. So, yes, I'm old. Back in the day, I was a hair and fiber examiner, trace evidence background, but also DNA. DNA, for touch DNA, for trace DNA, is another form of trace evidence, so it follows the same kinds of philosophy. So this bullpen of events recognize every alternative scenario is a single of series events, a series of single events. So this allows us to put together those independent events so you can come up with essentially any kind of scenario. So I'm going to talk about 7 new theories that have been borne out by this particular research that we're doing. So first, of course, is not ours. That's Locard's exchange principle. Every contact leaves a trace. So I'm going to talk about the 7 new theories that we're going to present here today. So the first one is, as Ashley has talked about it, we're calling it the theory of diminishing returns. That means with every new contact, you lose DNA. Not 100% of the DNA goes along for the ride. So, and it makes sense. Every time you touch a new service, every time there's a new transaction, you don't have the same amount of DNA. It's never 100%. So you always get less DNA, hence the theory of diminishing returns. The second one, preservation of mixture mass ratio. So when you have DNA, typically it's not naked DNA, it's bound up with cellular debris or it's inside cells. Those cells are there typically not in single source, but in some kind of a ratio. That ratio holds throughout this transfer. So if you have a 4-to-1 ratio of male to female, that 4-to-1 ratio for each other will hold. Now, other DNA can be entered, but the 4-to-1 in of itself stays the same, as in DNA doesn't jump out and clean itself off. Like an allelic ladder, the alleles don't take it off. Third theory is the last handler, as shown by our data, is likely their DNA is coming to them through that direct transfer. The last handler is direct transfer. In a series of events, the previous handlers are indirect transfer. Through that theory of diminishing returns, less and less DNA comes along for the ride. Most DNA is contributed by the direct transfer. So our experiments have actually showed the last handler as a direct contributor contributed 2.4 to 17 or to 15 times more DNA and 3 to 19 times more likely on the 3 substrates that we tested. And in the Idaho experiment, only 9% of the DNA was due to indirect transfer compared to direct. So 11 times more DNA is transferred directly. Hence, the last handler is likely the major contributor. Philosophy number 4, DNA is not a glitter bomb. So DNA is not self-mobile. DNA requires vectors for transmission. DNA just doesn't enter the room and poof, go everywhere in the room. There needs to be a deposit on original substrate. There needs to be a vector for transfer, and that vector deposits the DNA on a new substrate. every transfer is a separate and independent event, you need to have an explanation as to why the DNA got there. The fifth philosophy, every contact establishes a new equilibrium. So when you have a new contributor, they get added to whatever that mixture ratio is, and they dilute that. So if you have a mixture hand to substrate, the new person will be seen typically in that mixture. When new DNA or cells are contacted, the new transfer cells add the new complex mixture, which moves us into mixture number 6, one of my favorite, primordial soup mixture. So essentially, when you think about this room, how many people's DNA do you think would be actually present in the room? So it goes to the philosophy of entropy or mass chaos theory, but single source DNA is a rare event. It takes a lot of energy to maintain, but as soon as your DNA leaves your body, it goes increasingly into a more and more complicated mixture. And unlike fibers or hair where you have a microscopic capability of looking for, say, a red cotton fiber and picking it out, or looking for a hair that shares characteristics and cleaning it off, you can't do that with DNA. So there's no visual criteria. So essentially, mixtures become increasingly complex. You can't pick out the DNA and clean it off. And then the last one, which I alluded to, is DNA is not self-cleaning. Once it's in the mixture, the DNA does not become a single source DNA. It's inanimate. It doesn't climb off out of a mixture. So once you get that mass mixture, the soup mixture, it's not cleaning itself off. So that's what our research has borne out. And just to summarize, we've looked at the most favorable transfer experiments, and they've shown 2.4 to 15 times more DNA, in particular in this Idaho experiment with the SNP, 11 times more likely, more DNA is transferred. So, as I said, those substrates were free of contaminated DNA, and then just the idea that the incorrect suspect would need to touch exactly that spot or shake that hand. And between the two scenarios, my opinion is direct DNA transfer is more likely. So, sorry, my little sense of humor. This is not rocket science. It's forensic molecular biology. So by offering an opinion, is it unbiased or is it doing a disservice to the case? So certainly, if we don't opine, the jury, the finder of fact, is left without scientific opinion, and that is left to lawyers who are stating the case, as opposed to the science, which we have data to help you form that opinion. So withholding common sense opinion is not only unbiased, it's compromising, not unbiased, it is compromising justice. So with that.
Right. We have built a sound and robust model that permits evaluation of different events based on DNA recovery. It provides empirical evidence to inform activity level propositions and supports our 7 new theories. That is diminishing returns, preservation of mixture mass ratio, last handler is likely the major contributor, DNA is not a glitter bomb, each contact establishes a new equilibrium, the primordial soup mixture, I like that one too, and DNA is not self-cleaning. So juries are the finder of fact, they need guidance, and the forensic scientist is the best person to provide that guidance based on their experience and based on empirical data. In fact, they should offer their opinions as part of their testimony. Thank you for your attention. Please contact us. One more slide. Wait. We have to, of course, thank NIJ for funding and also the California Association of Criminalists. I have to mention Aldrin Alvear, who is the person who did all this work, and the DNA lab in Richmond was lucky enough to snap him up. My collaborator, Nick Miller, who helps with statistics and such, and, of course,
Thank you.
Thank you all.
Thank you.
We have time for a question or two. He's coming, he's right behind you.
There's a book by Malcolm Gladwell called Revenge of the Tipping Point, in which he points out that over 80% of the COVID spread was caused only by 10% of the individuals. There's certain people that are super spreaders of COVID. In our laboratory, we see that there are certain people who are super spreaders of DNA. Have you been able to look at, I see that I congratulate you on your experiments, but I'm curious about if these conclusions would work in the presence of a super spreader, for example, someone who constantly touches their face, touches their hair, or similar situations. And does the amount of the numbers that you have, are they just 1 nanogram, or would they work for 5 nanograms, 2 nanograms? Do they work for DNA that comes mainly from saliva or DNA that might come from sweat?
I can answer that. In fact,
It's very complicated if you have a situation where only a very few % of the people may provide a lot of the DNA.
no, it is, and this is why we've done 20 replicates. We did a power analysis to determine that that is the number that we need based on previous data from this. Yes, we have had, we started by experimenting with different concentrations. We've run a 5-point standard curve from 0 to 9 nanograms going into the reaction. We get the same results from all of them proportionally, so there is no concentration-specific effects. We've tested all.
Bruce, can you use the microphone, please?
Can you please reiterate what you're saying into the microphone, please? We have online audience.
So you were asking about super spreaders, and I happen to know, yes, we did have some included in that experiment. As you might guess, most of the DNA donated comes from people in my lab. And because most everybody works on this sort of thing, we know who sheds well and who does not. So yes, in fact, there are some people who shed quite well included in this. This is not one person's DNA.
No, my question is a priori. If you had such a person that was at the particular crime, it might look like that was a direct transfer simply because they had 8, 10 times more DNA on their finger than a typical person.
Mm-hmm. That's the question. Mm-hmm. Yes.
So I can throw in 2 things. So one is have a look at the article, because we've got a standard curve which is incredibly linear. So that's the beauty. The other thing is we now have the standardized fingerprint and mechanisms that you can follow exactly the same procedure, try out your different hypotheses with actual data, and be able to come back and say, in your lab, here's what you've been able to do. So certainly encourage repetition. More replicates, more scenarios are always great.
Great, thank you so much.
Thank you.
A round of applause. Our next speaker is Courtney Cavagnino. She is a research scientist at the Armed Forces DNA Identification Laboratory in the Emerging Technologies section. Thanks, Courtney.
Okay, so today I will be presenting on a comparison of Small-Amplicon mitogenome enrichment methods for massively parallel sequencing of low and high quality sample types. So my standard government disclaimer, and just noting that this work was funded by NIJ. So just to provide a little bit of background before diving into the study, massively parallel sequencing of mitochondrial DNA is critical in historical and forensic cases involving degraded remains and or distant maternal relatives, especially in the work that we perform at AFDIL. AFDIL validated in 2016 a PCR enrichment method with 2 long-range amplicons that are about 8.5 kilobases each. And we use this method for population databasing and reference sample processing. And then on the other end of the spectrum, we validated a mitochondrial genome or mitogenome hybridization capture method in 2016 as well. So this method utilizes baits or probes to target our desired DNA type. And this allows us to decrease the bacterial DNA content within a sample. And this method is amenable to DNA fragments that are less than 100 base pairs and are highly degraded. But what about the samples that fall in between the long-range PCR method that are high-quality reference type samples and our highly degraded samples? These samples can also benefit from the efficiencies of NGS processing, and that's what we look to evaluate in this study. So the goals of this study are to evaluate these commercial Small-Amplicon mitogenome enrichment kits for MPS. And these kits have amplicon sizes between 100 to 250 base pairs. And we're looking at evaluating these for both casework and reference type samples. And in AFDIL, this is the goal to ultimately replace our Sanger processing. And then for the larger forensic community, we want to assess the overall performance, processing time, and cost of these kits to assist forensic practitioners looking to implement this technology in their own laboratories. So overall, we processed 96 samples, and where applicable, I generated replicate extracts and homogenized them for use across all of the kits to be able to eliminate that as a variable. And across all of these samples, we had a variety of sample qualities, sample types, substrates, extraction methods, and mitochondrial haplogroups. So we incorporated no template controls, control DNA, mixtures, reference swabs, blood stain cards, NIST research grade test material, non-human samples, bone, hair. We had 3 samples that were created into serial dilutions, and we had a set of population samples. And including all these samples in these studies, we were able to address a number of internal validation studies in accordance with FBI QAS standard 8.3. So some of these studies included known and non-probative evidence samples, sensitivity, mixtures, and contamination. And the remaining study that would need to be completed is precision and accuracy. And we also addressed species specificity during this testing. So just some brief background on Small-Amplicon MPS kits in general. These kits utilize small overlapping amplicons for complete mitogenome coverage, as you can see in the general image on the right side of the screen. And the benefits of these kits are that they're cost-effective, quick, they have amplicon sizes that are comparable to primer sets typically used for Sanger processing, and most have an associated analysis software. However, there are some known complications. So some of these kits run into primer binding site issues due to mutations. However, this can be overcome due to the incorporation of degenerate primers into the panel design. Additionally, another known complication is the co-amplification of nuclear mitochondrial DNA segments, otherwise known as numites, and these can be overcome with stringent bioinformatic alignment strategies during the analysis protocol. So the 5 kits that we evaluated were the ID-Seq mitochondrial DNA full genome sequencing kit by Nemogen, the QIAseq targeted DNA human mitochondrial panel by QIAGEN, Precision ID mitochondrial DNA whole genome panel by Thermo Fisher Scientific, the PowerSeq WGM system by Promega, and the Forenseq mitochondrial DNA whole genome kit by Verogen QIAGEN. So now I'm just going to go into a bit of information regarding each of these kits. So for ID-Seq, the DNA input recommendation is 100 picograms in total, so that's 50 picograms into each of 2 pools. This kit includes 100 amplicons, with the minimum amplicon size being 150 base pairs, the max 250, and the average 236. This average is on the higher end for all the kits that I'll be introducing. And for the analysis software, there wasn't a recommended software for this kit, so we utilized CLC Genomics Workbench, which is our default software for mitochondrial DNA NGS analysis at AFDIL. And some highlights, this kit utilizes reverse complement PCR, and following the initial PCR step, the panels are pooled, minimizing downstream processing. So with this protocol, you could ultimately end up with 1 tube per panel A, 1 tube per panel B, with all of your samples pulled together in each of those, continuing through the procedure. For QIAseq, the DNA input recommendation is 10 to 80 nanograms, so this is the highest of all the kits that I'll be discussing. There's only 1 PCR pool, and it consists of 220 primers. There are no amplicons for this kit. And the recommended analysis software is the CLC Genomics Workbench. Some of the highlights include that this kit utilizes unique molecular indexes, otherwise known as UMIs, that are ligated to the original DNA molecules. And additionally, this kit utilizes target enrichment through single primer extension, which can be beneficial for degraded remains. Precision ID, the recommended DNA input, is 100 picograms into both PCR pools, so 200 picograms in total. This kit has 162 amplicons, the minimum 120 base pairs, the maximum 185, and the average at 163 base pairs. The analysis software that we used for this kit was Converge. Some of the highlights include that this kit allows for automated processing, and there's smaller amplicon sizes. So this range shown here on the screen is smaller compared to some of the other kits. For PowerSeq, similar to many of the other kits, the DNA input recommendation is 50 picograms per PCR pool. Number of amplicons, 161, with a minimum amplicon size of 92 base pairs and a maximum of 254 base pairs. That average is 167. So here we have our widest range in amplicon sizes. And the recommended analysis software is GMarker HTS. The highlight for this kit is that there's only one PCR step, then the pools are combined for downstream processing, minimizing the hands-on work. And then finally, we have Forensic. So the DNA input recommendation, once again, is 50 picograms per pool. There are 245 amplicons. The minimum amplicon size is not advertised in the user's manual. However, the maximum is 209 base pairs, and the average is 131. So that's much lower in comparison to some of the other kits presented. And the recommended analysis software is the Universal Analysis Software, otherwise known as UAS. Some of the highlights for this kit include that Forensic is a well-established kit, and many laboratories have validated signature prep, so this could be an easy transition into using the Forensic whole mito kit. Additionally, UAS is a semi-automated analysis program that is user-friendly. So this screen here just shows a brief overview for each of these kits to allow you to compare the number of PCR pools for the kits, the number of amplicons, and the average amplicon size, just to kind of start getting an idea of how these kits may work based on your sample quality. And there's a lot on the screen here, but it's just a brief overview of all of the workflows for each of the kits. We followed the manufacturer's recommended protocols, including multiplexing levels for sequencing. For this, the only exception, though, is precision ID. We sequenced 16 samples where the manufacturer recommended 32. The DNA input was based on the quantifiler trio, small autosomal target for all of the kits. And just kind of looking at these protocols here, you can look at the number of PCR steps, the number of purifications, where the samples can be pooled or combined, just to kind of get an idea of how labor intensive each of these protocols may be. For the analysis, we utilized the default analysis software and parameters where applicable. As I already mentioned, for ID-Seq and QIAseq, we use CLC Genomics Workbench, Converge for Precision ID, GMarker HTS for PowerSeq, and UAS for Forensic. And in the table here, I included the minimum coverage values, the minimum variant count, and the minimum frequency. So looking at the minimum coverage, the lowest we have is QIAseq at 10, for Forensic, which is the highest at 45. For the minimum variant count, once again, QIAseq is the lowest at 2. Forensic is the highest at 45. And then looking at the minimum frequency, many of the kits utilize a minimum frequency of 5%, and Precision ID is the highest at 10%. For QIAseq, we are able to have lower analysis thresholds just due to the inclusion of UMIs, allowing you to analyze those original DNA molecules. Some additional processing that we performed was long-range PCR and hybridization capture. These are two routine methods that we perform at AFDIL, so we included these for baseline processing. And the haplotypes that were generated from each of these methods, we were able to use for haplotype concordance in comparison to the Small-Amplicon PCR kits. So for long-range PCR, for that testing, we didn't include the bone samples just because we knew they would fail based on the degraded fragment sizes. So that left us with 89 samples that were processed. For this kit, the input is 5 microliters per amplicon. And then we have that PCR step with the 2 amplicon sizes that are about 8.5 kilobases each. These samples undergo Kappa Hyper+ library preparation, and this is also an automated protocol. And these samples, we were able to sequence all of them on the MiSeq FGX cartridge. For the hybridization capture protocol, we only did a subset of samples, so we had 28 samples that were processed. And I just want to point out that the input for this method is based on qubit, so it's looking at total DNA. And the maximum input is 1 microgram into library preparation. For the low-quality samples, they underwent a repair step before going through the Kappa hyperprep protocol, and our higher quality samples went through HyperPlus. We analyzed both of these, the data generated from these methods in CLC Genomics Workbench. And then on the screen, you can see the minimum coverage, minimum variant count, and minimum variant frequency for each of these methods. So this chart here is the sensitivity results for each of the kit. So I only included the overlapping values across each of the kit, and that range from 15.6 picograms to 1 nanogram. For some of the methods, we did include inputs of 5 nanograms and 50 nanograms for those that had allowed for higher inputs. So to just get you oriented to the chart, on the left y-axis, represented by the columns, we have the average coverage. On the right y-axis, represented by the line, we have the percentage of bases covered across the mitogenome. And this data was generated from replicates of 3 controls, so 2 high-quality samples and 1 degraded sample. So what you may notice right off the bat here is for the long-range PCR data, ID-seq and chi-seq, you may notice that these kits had much lower average coverage values in comparison to the others. But this is largely due to the multiplexing levels. So for these 3 kits, we were able to sequence 89 to 96 samples in 1 sequencing run in comparison to the other 3 methods included in this chart. Taking those aside, PowerSeq and Precision ID had the higher average coverage values. But when you look at the percentage of bases covered in the mitogenome, you'll see that there wasn't an impact on that despite the differences in the average coverage. So looking forward now at the remainder of our samples that were included in the study, we have these grouped by no template controls, non-human samples, bone samples, hair, and high-quality samples. So this chart here just shows the average coverage. In each of these groupings, they take the average of the samples that fall into those categories for each kit. So once again, you'll see that for long-range PCR, ID-seq, and chi-seq, you are seeing those lower average coverage values in comparison to PowerSeq, for example. PowerSeq across each of the categories did have the higher average coverage values consistently, with the exception of the non-human sample, the non-human category, I'm sorry. There was 1 sample that was an outlier, which really drives up that value. But once again, when we look at the percentage of bases covered across the mitogenome, specifically at the hair and high-quality samples, we see that it didn't have an impact on the percentage of bases covered. And then interestingly, looking at the bone samples that are lower in quality and would have more variable fragment sizes, the hybridization capture and precision ID methods performed best in generating almost full mitogenome profiles. The haplotypes were concordant for overlapping regions between all methods, including long-range PCR and mitogenome capture, and all mixtures were correctly identified. There is a need to further investigate some low-level variants that we believe are due to new mites or artifacts based on the calling thresholds. Additionally, we performed a time and cost analysis. So we looked at batch size based on the maximum recommended multiplexing levels, except for precision ID and that caveat I gave before. And for chi-seq, we looked at 2 library preparations of 48 samples. The cost does not include the cost of consumables or quantification reagents, and time includes library preparation through data generation. So on this chart here, on the bottom left, we have the batch size. So I'll start here because that highly influences the cost per sample at the top, as well as the time and hours at the bottom right. So for batch size, as I mentioned a couple times now, ID-seq and chi-seq, we were able to process 96 samples at a time. And the smallest batch sizes were associated with precision ID and forensic at 16 samples. Then looking at the cost per sample, forensic was the most expensive, and ID-seq was the least expensive at less than $80 per sample. So you can kind of start to see the influence of the batch size there. And then when you look at the time and hours, PowerSeq, ID-seq, and chi-seq appear to take the longest. However, it's really important to factor in the batch size when you're evaluating this parameter, because it does appear that forensic and precision ID take the least amount of time, but that's only for processing a set of 16 samples. So you would need to multiply that time by 6 to get a true estimate of how long it would take to process 96 samples, and that would take you beyond what you see for ID-seq and QIAseq. So hopefully if there's any laboratories that are looking to implement any of these methods, hopefully the slide can provide some insight and help make your decision. Oh, I'm totally hitting the laser. So some overall conclusions after completing this work. Small-Amplicon kits may be necessary for even high-quality reference type samples when long-range PCR fails. Average coverage is largely influenced by multiplexing levels. Greater than or equal to 98% of the bases were covered for all kits, down to 15.6 picograms of high-quality DNA input. And all kits produce relatively comparable results. So laboratories will need to assess the factors such as time, cost, amplicon size within the context of their intended applications. So now just some conclusions on each of the kits, some pros and cons. ID-seq utilizes reverse complement PCR, target enrichment, and indexing within that first step, so it really minimizes the potential for contamination. And it has the lowest associated cost per sample. And something that may be seen as a pro or a con, based on what you currently utilize in your laboratory, is that there is no associated analysis software. QIAseq utilizes UMIs to ensure only the unique reads are represented in the data, and it incorporates target enrichment through single primer extension, which is beneficial for degraded remains. However, QIAseq is labor-intensive with many hands-on lab steps, and it has the highest DNA input recommendation. However, based on the sensitivity data, we did see that you were able to generate almost full mitochondrial DNA profiles down to 15.6 picograms of high-quality DNA. For the precision ID, it allows for automated processing on the Ion Chef, and it does have a smaller amplicon size range, which we saw was beneficial for the bone samples that were included. And it performed, which is right there. For PowerSeq, only one PCR step, then the pools are combined, so that really minimizes the downstream processing. And it had the smallest minimum amplicon size. It also has an associated analysis software with an established workflow. And looking back at all these kits, we said this kit was overall very average. For forensic, it's a well-established kit. However, it has the highest associated cost per sample. And UAS software is semi-automated and user-friendly. So depending on what analysis software you use now and how much information you're used to seeing, this could be a benefit or a con. In the future, we plan to test samples with precision ID amplification and downstream kappa hyperprep library preparation to allow for Illumina sequencing. And we also need to complete the analysis of NextSeq and NovaSeq data for long-range PCR, mitogenome capture, and whole genome sequence data. And then we also plan to analyze these data sets across multiple analysis softwares, including GMarker HTS and CLC Genomics Workbench, and to optimize those analysis workflows. In conclusion, I would like to thank Dr. Jennifer Solar, Dr. Michael Coble, and the UNT HSC team for their work with the precision ID processing and their help throughout the project. I would like to thank the rest of the Emerging Technologies section at AFDIL for their support, April Higgins from SNA, and Dr. Timothy McMahon from AFDIL for logistical support, NIJ Forensic Science R&D Symposium Organizing Committee for allowing me to present here today, and NIJ for funding. And I will take any questions.
Yeah, Tracy?
I have my own personal question. Courtney, can you talk a little bit about homopolymeric regions and how the software handles like insertions, deletions?
So it's definitely dependent on the software that's used. There are some differences. For example, the forensic, it does have some issues with insertions and alignment, and I'm currently in contact with QIAGEN to determine how to call insertions in the software that may not be there in order to shift them. So that's one issue we're currently having. In GMarker HTS, I also did have some trouble with alignment, so those are things to look into.
All All right, see no other questions. Let's thank Courtney. All right, and so our next speaker is Samuel Sacco. He is a PhD student in Ed Green's Paleogenomics Laboratory at the University of California, Santa Cruz. I will pass it over to you.
All right, so today I'm going to be talking about kind of a newer avenue of research that my lab's been exploring, and that's how to actually ascertain the epigenetic profile of degraded sample types like rootless hairs. This is your standard NIJ disclaimer. Obviously, this doesn't reflect the opinions of the NIJ. And so I guess I want to start with kind of why rootless hairs are interesting. I think it really kind of comes down to how ubiquitous they are as a source of evidence. We all shed between 50 and 100 hairs daily. These hairs are kind of in the final stage of that growth cycle. It's called the telogen phase, where the root recedes, and you're just left with the hair shaft itself. And for a long time, it was kind of thought in the field of forensics that there was no DNA that's retained in the hair shaft, right? It's all kind of in that root. But in the last 10, 15 years, there's been some studies that have shown that there actually is DNA present in the shaft. And so this is a nice kind of fluorescent microscopy image showing the retention of nuclear remains in the hair shaft. You can see it varies dramatically between individuals. What you can actually do is do a DNA extraction, isolate the DNA from those rootless hairs, and then sequence it and see kind of how long the fragments you get are. And that's what this length distribution is showing. And you get this really interesting sawtooth pattern where there's this 10 base pair periodicity. That has to do with how the DNA is actually kind of wrapped around the nucleosomes. But really, I just want to point out that that DNA is present, but it's extremely fragmented, right? So it's only 30 to 50 base pairs on average. That's 30 to 50 letters. And when you compare that to like a typical STR marker, which are usually 100 base pairs or larger, or a high-quality DNA sample like a spit sample, you're often dealing with thousands of base pairs of DNA. But we can take a forensic sample like rootless hair. We can get the sequence data out of it, and then from that sequence data, we can actually do this new genotyping approach, right, where you're kind of saying what the different variants are in this person's genome. And once you have that, you can kind of take that genotype file and do a couple of things. You can query it against a large database like GEDmatch and say, hey, are there any relatives, or can we kind of do this sort of genealogical approach? Or often you'll have a high-quality DNA sample from somebody, and you can do sort of a direct comparison. But oftentimes when you do this, you get no match, or perhaps you only identify a distant relative, and you have too many relatives to really search through all of them. And so we kind of wanted to think, if we can get this sequence data from the hair, what else can we get? What other sort of useful biological information can we ascertain from a rootless hair? And so I think that's where epigenetics can really be useful. So a lot of times when people are thinking about epigenetics, the typical sort of research workflow is you start with these different environmental conditions, different, environmental in the large sense, the way genetics or geneticists think about the environment. So things like age or disease or exposure to toxins. And then you ask, how do these environmental conditions actually lead to physical changes in our genome or chemical changes? And so here I'm showing kind of one of the most common. When people are talking about epigenetics, often they're talking about cytosine methylation, where you get this little chemical group added to those C bases in your genome. And this tends to happen at places called CPGs, which is really just kind of a fancy word for saying places where you have a C followed by a G. So instead of doing that typical epigenetics pipeline, we can kind of flip it and start with measuring the epigenetic changes in a forensic sample. So we get a rootless hair or whatever, and we assay where are these cytosines methylated. And then using our understanding of how environmental conditions create these changes, we can sort of reconstruct the environment. And here for this talk, I really want to focus on age. This is something that's super well established in terms of we know very clearly that as you age, you undergo these sort of characteristic changes to your CPG methylation. And then once you have age, there's sort of these natural forensic applications and That could just be estimating somebody who left something at a crime scene, or perhaps you have somebody but you don't know how old they are, and so you can estimate their true age. And to really motivate this, I want to talk about kind of an older study that showed if you have methylation values of just 350 of these CpGs, you can actually predict somebody's age to within 3.5 years. And so this was over 10 years ago now, but even in this paper, they acknowledged that this had a direct forensic application. And since then, a ton of people have come out in the forensics community and tried to sort of adapt this and make it work for different tissue types, and really make it sort of something that would actually be useful to practicing forensic scientists. And so there's been a lot of great work, but I want to caveat that it's only feasible in certain tissues that can yield high-quality DNA, and that it requires these very specialized assays to estimate that methylation, right? So it's not something that you can simply do in a normal forensics lab. This is sort of a very sort of specialized area of research. And right now there are two kind of main approaches. By far, what people do to estimate cytosine methylation is called bisulfite sequencing. This is where you sort of wash your DNA in this chemical called sodium bisulfite. It causes hydrolysis of those methyl groups, and then in subsequent sequencing, you can kind of tease out which of the cytosines kind of had that methyl group and which did not. But you can imagine if you're washing your DNA in a hydrolyzing chemical, it's going to cause fragmentation of the backbone at some level. And so in a sample where the DNA is already highly fragmented, that's no good. You're going to turn your DNA unusable, and it's going to prevent you from sort of actually generating those genotypes, which you also want in the first place. And generally, these bisulfite methods require large inputs of DNA. In a separate category, there are these long-read methods. They're sort of newer, but the name kind of gives it away in that your DNA has to be high molecular weight. It has to be very long. And so for now, we're kind of living in this short-read world. And so in the field of liquid biopsy, there's been a really kind of interesting new approach for estimating cytosine methylation. And it turns out that what people have found looking at DNA from blood is that when that C is methylated, when it has that little chemical group, it's almost like it's saying cut here, right? And so you'll observe lots more fragmentation occurring right at that C position relative to the adjacent positions, right? And this study was really cool. It's a totally novel way of estimating this methylation. You can imagine it's good for samples that are already fragmented. But there was a few caveats with this study as well, mainly that they only looked in blood, so maybe that's good if your sample's a blood sample, but for hairs and other things, they don't know if this pattern existed in that. And then also, they only looked at one end of the DNA molecule, right? So they only looked at what's called the 5' end, but you can imagine if that break's occurring, it's creating two ends. It's creating a 5', and then on the other side, it's creating a 3' end. And so we wanted to see, can we take this approach and apply it to hairs using both the 5' and 3', so try and kind of add, hopefully, a little extra data. And so this is what it looks like in blood plasma. This is just showing, again, more results from that study. And what you see is, at unmethylated CpGs, that blue line, as compared to methylated CpGs, the red line, you have about 3 times less cleavage right at that C position. And so it's a very strong signal in blood plasma, but again, no one knows how this looks like in hair. And so this was sort of our approach. We had hair DNA from a large panel of individuals that we collected, and we looked at where did those 5' and 3' termini occur relative to CpGs that we knew were methylated or unmethylated from previous experiments looking at hair follicles. And when you do this, fortunately for us and my PhD, you see a really strong signal where the cleavage is elevated, almost more so in hair relative to blood plasma, right? And also good news is that we see it not just on the 5' end, that top row, but we see it on the 3' end as well, right? And again here, red is the methylated CpGs, and blue is the unmethylated. And so this is great. It's establishing that that signal exists, and this might be useful if you're wanting to ascertain methylation in fragmented sample types. But we wanted to take it another step further. I'll start by saying, we wanted to kind of take it another step further and really fully explore, I guess, the landscape of informative termini around a CpG, right? So that previous study was just saying, okay, you have reads that start at C or start away from C, but we can actually ask, what are the specific correlations of observing termini at different positions around that CPG, right? So this solid blue line, for instance, is looking on the forward strand and looking at the 5' ends. And again, we see there's a strong correlation between that CPG being methylated with observing those forward 5' read starts. And then dashed lines are the 3' end. So again, it's kind of that same fragmentation event creates both the 5' and the 3'. So we can see that those are also useful. So this is important if we want to develop a modeling approach. And then one other thing I want to point out is that the forward and reverse strands, the orange and blue lines, are perfectly symmetrical. So this is kind of what we would expect. Biologically, there's not really a difference between the forward and reverse strands in terms of what an enzyme is going to cut. And one last thing, I guess, is that the rootless hair profile looks very different from the blood plasma, which is also interesting biologically because it may mean there's sort of different enzymes involved or a different process creating the signal. And so now I wanted to get into actually using that information that we've established exists to actually modeling CPG methylation just from termini, right? And if we could do this, it would be great because you just sequence your DNA, you do no additional assay, and if you have enough information to accurately model that methylation, you just need a good computer program, basically, to get back to these interesting environmental variables like age, right? So here, we took those 5' and 3' termini, we plugged them in as features of a really basic, just logistic regression model, and we saw how well were we able to predict CPG methylation just based on observing those features, right? And so on the y-axis of this heat map is our prediction, and on the x is the methylation as we ascertained it in a different experiment. And we have a pretty good correlation overall of about 0.71 using this extremely simple approach, right? But going through this research, we kind of made some other interesting observations, which is that when you change the local sequence context, otherwise, like what bases are around that CPG, it drastically changes this sort of fingerprint of correlations, and it changes what features are going to be important for modeling, right? And so you can imagine, I mean, if you look at the top row, for instance, as compared to the bottom row, you'll see that GC, CG, GC has a very different profile than CA, CG, TG. And so if we lump these into one model, and that logistic regression has to learn the weights, it's just going to kind of average everything out, and you're going to lose a lot of sort of information that's present. And so we tested sort of a second approach. Again, we're measuring 5' and 3' termini. But this time, we're kind of creating a separate model for each sequence context, right? So there's a different logistic regression model depending on what those 4 adjacent bases are to the CPG. And doing this, we actually get these really sort of higher accuracy models, correlation of around 0.82. So we're pretty happy with this, but there is a sort of a few caveats with our experiment. And really, all of this has been done in sort of a validation data set that's not really reflective of what a true forensic sample might be, right? So we're working with hundreds of termini being observed in these windows because we've kind of combined data from many, many hairs into one data set, right? And so actually moving this to something that would be useful for a forensic scientist, we have to kind of scale the model down and give it less information to work with and then still make those accurate predictions, right? But unfortunately, the model's performance, it degrades pretty substantially once you get below 50 termini observed, right? And so sort of a general caveat, but I want to say that doing it this way is sort of a really kind of hard problem to address, right? We're trying to estimate methylation at every one of 28 million CPGs in your genome using that fragmentation profile. But when we think about an environmental variable like age, it changes CPG methylation in a very sort of global fashion, right? And so there should be a way to sort of combine these different CPGs and aggregate that data, so aggregate at the CPG level, and then we can kind of get back to our accurate prediction by sort of combining the information from multiple CPGs. And so that's sort of our next steps. And then, again, a nice conclusion of this, which hopefully is useful for us and for people in the liquid biopsy field doing this in blood, is that using the 5' and 3' end is important, right? You get most of the information from the 5', but that 3' end really does help you a little bit, right? And with this sort of thing, you kind of want every little piece of help you can get. And so, yeah. So kind of moving into to my conclusions, the big thing I kind of want you all to walk away from is that there's sort of this unique way of estimating CpG methylation integrated sample types just based on fragmentation. And that actually will facilitate a modeling approach just from regular shotgun sequencing data, right? So no additional assays are required. You just sequence your hair, you generate your genotypes, or perhaps a different sample type, but you generate your genotypes from your degraded sample, and then that information that was present, or just those termini are present immediately in that sequencing data, and you can use them to sort of also ascertain the epigenetic profile, right? And so going forward, we're looking to sort of use this information to develop sort of more complex models to actually estimate how old someone is, and hopefully scaling it down to just a single rootless hair. And so there were a lot of great people that helped me with this. Ed Green, Josh, and Remy are all my little forensics team at the University of California, Santa Cruz. The NSF for supporting me, the NIJ for supporting me and letting me talk here. And yeah, feel free to contact me or meet me after the talk or anything.
We have plenty of time for questions.
I have my own personal question, Sam.
Sure.
Oftentimes, forensic science has really borrowed from other sort of communities, the medical community, the paleogenetic community. This is kind of the opposite, I see. It's almost like this sort of forensic question has created this opportunity to understand fundamental biology. Would you agree?
Yeah, I think this is definitely something we wouldn't have explored if there hadn't been that forensic motivation. And our lab sort of pivoted towards working on this kind of after we had already started doing more traditional sort of forensics to estimate the genotypes from rootless hairs, right? So this was sort of like a side thing that spun off of that. And really, if you look at like the data on this grant, it's like 2020. That's what the grant was for, but we've kind of since moved into this, and now we're starting a new project on it. So yeah, it really kind of was born out of wanting to address this forensic question specifically.
Really fascinating stuff. I guess my question is, when you were looking at the bases like adjacent to the CpG islands, was that something that you expected, and how did you decide to pick 4 as like the number, I guess?
Yeah, we picked 4 because it was a convenient, like the number of combinations once you get past 4 kind of scales extremely dramatically, and then we're reducing our data set of CpGs kind of down to smaller and smaller amounts, and we can't really accurately estimate what the correlations are. So that's kind of why we picked 4. It's sort of a limitation of that. I guess as far as expectations, there is this expectation that this signal is generated enzymatically, and different enzymes have sort of different base preferences. So we kind of went into it maybe thinking there would be a difference, but to see it so drastically in hair and in the blood plasma was definitely a surprise, for sure.
Any more questions? Tracy, anything else online?
No.
All right.
All right, let's give a round of applause. And we have plenty of time, but according to our agenda, so we'll see how long I can talk up here. Okay, so our next speaker is Katherine McBroom Henson. She is a graduate student at the University of North Texas Health Science Center, and she is completing her PhD in biomedical sciences. And with her research, she wants to blend genetics with anthropology. So that is a little bit of what she will be talking about today. And I got 2 minutes down, but we're still 5 minutes ahead. So sorry to those online. But Katherine, you're more than welcome to come up here. That means I expect lots of questions today.
Okay. Cool. Can everybody hear me from this position? Okay, cool. So my name is Katherine McBroom Henson, and thank you for the wonderful introduction. And today I'll be giving a brief update over my current work, which is entitled Adaptive Sampling for the Simultaneous Analysis of STRs, SNPs, and Mitochondrial DNA in Human Remains Identification. Okay, standard disclaimer we've heard all day, the comments, the opinions, the recommendations are all mine and not that of the Department of Justice. And I wanted to start my talk today by giving a brief overview of the technology that I'm using. And so my lab focuses on adapting Oxford nanopore technology for forensic applicability. And this all started in 1989 with David Deamer, who first conceptualized Oxford nanopore technology. He believed that a nanopore could be embedded in a membrane and that individual nucleotides could be accommodated within that nanopore to be able to be sequenced. And after years and years of proving that idea and developing that idea, Oxford Nanopore was founded in 2005. And so in 2012, after 7 more years of research and development, which really goes to show how important research and development is, the first DNA sequencing data with a nanopore was presented. And 2 years later, the MinION Access Program was allowed. And this was an early access program where participants could get the MinION technology and kind of do their own research prior to commercialization, which began in 2015. And then from 2015 to 2019 really was just a lot of further development, further research, further improvements on the technology itself. And in 2020, at the height of the COVID-19 pandemic, adaptive sampling technology was released. And this is a targeted sequencing approach that we'll see could have some implications and use in forensics. And so lastly, from 2021 on to present, Oxford Nanopore has focused on developing higher accuracy with their long reads data, as well as developing kits to support the accessibility and broad applicability of this technology. And so how does nanopore sequencing work? So embedded in this array are a bunch of tiny little nanopores in a membrane, and that a membrane has an ionic current run through it. The ionic current will be disrupted as the nucleotides are shoved through the nanopore and read by a reader. That creates a characteristic squiggle, and that is a technical term, which is read and measured by the application-specific integrated circuit, and later can be base called into your individual nucleotides again. After base calling, any number of downstream analysis can occur, including what we do normally. And so because adaptive sampling and nanopore technology runs on a voltage, that voltage can be reversed and the nanopore can spit out the DNA that it was currently sequencing. And so adaptive sampling works as an in silico sample manipulation method for targeted sequencing of your regions of interest. And it comes in 2 flavors, enrichment mode and depletion mode. Enrichment mode preferentially selects your regions of interest, which you provide the Minnow software, and rejects everything else. Whereas depletion mode ends with the same goal, but it's a negative selection of regions you don't want selected. So you say, I don't want A, B, and C, and it does not sequence A, B, and C. And to do this, you require a bed file containing your regions of interest for enrichment mode, or containing the regions that are not of interest for depletion mode, and a FASTA file that matches that reference. So a little bit more detail about how adaptive sampling actually works. Within that bed file will not only be your regions of interest, and just real quick, as a note, this is all for enrichment from here on out, just to simplify things and make it easier. So in that bed file, you have your regions of interest, and you're going to add a 10 kilobase pair buffer to the end and the beginning of your regions of interest, and that ensures that any fragment that might not be within but will lead to your region of interest, like in panel B at the bottom, will continue to be accepted for sequencing. The sequencing platform will read the first 400 bases, which will be base called and aligned to the reference file, and then subsetted across your bed file to determine if it is or is not a region that should be sequenced. And then best practice for this process is that the live alignment and the live base calling features for nanopore technology is turned off. Now that might sound counterintuitive because I just told you that we have to base call and we have to align our fragment. Now the base caller and the aligner for adaptive sampling are actually separate to what is used in the live alignment and line base calling features in Oxford Nanopore Technologies. So, since its release in 2020, adaptive sampling has gotten some action, but it's recently picked up quite a bit and has been used in metagenomics, pharmacogenomics, clinical diagnostics, and forensics. And I feel like the applicability of this technology in forensics is pretty clear. It's limited sample manipulation, at least in terms of wet lab surfaces. So there's low possibility for contamination, sorry. It's been a long day. And it could also be portable, which brings me to the overarching research aim of our whole project, which is, again, to evaluate the forensic applicability of the newest and cheapest NGS platform, ONT. So some of the advantages of Oxford Nanopore technology is that it is portable. Many of its device options plug into a laptop or a PC computer, such as the MinION or the P2 Solo, and they've recently released a commercialized version that stands alone. So it combines the computing power and the sequencing power into one device called the Prometheon 2 Integrated, which is what I used in this project. It also contains a relatively low startup cost, and so the potential for on-site sample processing, given this portability and low cost, is clear. So the goal of my study was to design a single assay for the evaluation of short tandem repeats, single nucleotide polymorphisms, and mitochondrial DNA for the purpose of human remains identification. So how did we go about doing this? We started with sample processing. Obviously, as we know as forensic biologists, a good library prep will hopefully lead to good data. And so we used the ligation sequencing kit for genomic DNA of 96 barcodes. And I want to make that note because it does matter later. We used an input of 100 nanograms, but our final library resulted in only 3.4 nanograms, which means we lost about 96% of our sample during the library preparation process, which was considerable. So in talks with ONT and through some optimizations of ourselves, we did optimize this protocol. We switched from the 96 native barcode kit to the 24 native barcode kit. Even though they're the same kit and the only difference is the amount of barcodes you receive, the protocols do have slight differences. And so making this switch improves our yield and DNA retention. So with our basic modifications, which is really just extended incubation times and high resolution temperatures, we've had an input of 2.2 nanograms and a final library of 0.92 nanograms. So we were able to retain more than half of our DNA library and only lost about 58%. So overall, our basic modifications were able to improve yield, and I wanted to walk you through not only the original protocol, but also the modifications that we made. So, in short, the original protocol, let me go back actually, because this is a better picture for it. The original protocol has high molecular weight DNA that, if necessary, is fragmented, and then it's DNA repair and end-prepped. You ligate on barcodes and adapter sequences, and then you load the library onto the flow cell for actual sequencing. Perfect. Okay. So going into the modifications that we made, in fragmentation, we reduced the fragmenting volume by 2/3, and we added an ampere bead cleanup. Now, this bead cleanup can be done to elute into nuclease-free water rather than having any kind of elution buffer that may be altered in terms of concentration when you eventually vacufuge down to 12 microliters so that you can input into that DNA in prep and repair step. In this step, you usually use DNA control sample as a troubleshooting purpose, but instead we omit that and we make it up using our DNA sample itself. We also extend the incubation times for this reactions to maximize the amount of DNA that is repaired, and we modify the ampere bead cleanup to retain DNA as much as possible. During barcode ligation, again, we vacufuge down to the input volume, and during the pooling process, we carry over the entire sample volume rather than carrying over only by mass. And we do, again, that modified ampere bead cleanup to retain as much DNA as possible. We move into adapter ligation, which from barcode ligation to adapter ligation, there's no need to do another vacufuge step. You'll elute directly into that input volume, but you do extend the incubation time so that as many ends of DNA can have those adapters ligated onto them as possible. It's required as the motor protein for sequencing is tagged onto that end of the adapter sequence, and so in order for your DNA to actually get sequenced, you have to have the adapter on there so the motor protein can bring it to the nanopore. Lastly, again, we do that modified ampere bead cleanup, and this is again further modified by using, instead of ethanol, the short fragment buffer that comes with these kits, again to maximize DNA retention and yield. Lastly, during flow cell priming and loading, you use library beads rather than library solutions, which are for more viscous libraries, and you wait 1 hour prior to starting your sequencing to maximize your data output. So in summary, the original protocol only takes about 2.5 hours, and our modified protocol takes about 12 hours, and that can be seen as a major downside, but in terms of the DNA retention and yield, it is considerably different. Okay. And then just to summarize again, you vacufuge to input volumes, you extend your incubation times, and you change your Ampere bead cleanups to retain as much DNA as possible. And so when we use this protocol, we multiplex 3 samples of 2800 M controlled DNA together. 2 samples had 250 nanograms, and the last sample had 90 nanograms. And our final library was 151.8 nanograms total. And I want to note that we sheared this controlled DNA to approximately 15 kilobase pairs to try to build a mock forensic cane sample. Thank you. Okay, so the adaptive sampling parameters were as follows. On a Prometheon 2 integrated, we had a runtime of 72 hours, and we did use enrichment mode, which, as a reminder, preferentially selects for the regions of interest that we want sequenced. Our regions of interest included 71 STRs, greater than 5,400 SNPs spanning kinship, ancestry, phenotypic informative SNPs, as well as X and Y chromosome SNPs, as well as methylation loci. We had an indirect enrichment of mitochondrial DNA, meaning we did not put mitochondrial DNA into the bed file itself. We believed that due to the high copy number in each cell, mitochondrial DNA would come back fine in the background. And while I don't have the data in the presentation, just as a note, it did. We also had that 10 kilobase pair buffer region at the start and at the end of each of our regions of interest to ensure that fragments leading up to our regions of interest would get sequenced. And we matched our bed file to human genome 38. So doing data analysis, you use the Dorado base caller to take your initial squiggle data and bring it into nucleotide sequence data. That gives you an unaligned BAM file, which you'll then put into Minimap2 to align. And then lastly, you take your aligned BAM file, and we got just preliminary data for right now, but just coverage data using QualiMap and a BAM-QC. So here's our run statistics, and ultimately what we discovered was that pore activity fell quickly, and this wasn't a shock. With adaptive sampling, as you might suspect, a lot of your nanopores go empty, and when your pores are left empty for a long amount of time, like 72 hours, they collapse in on themselves. And when they collapse, they cannot be uncollapsed. And so that reduces the flow cell efficiency and reduces your data output. This can be resolved by doing flow cell washes and resequencing or reloading of your library to re-engage and refuel the flow cells, or the nanopores that are still available. But adaptive sampling was active. So one thing that I forgot to mention earlier is that adaptive sampling will have an N50 or a median fragment length of anywhere from 400 to 600 bases. And what we found was our N50 fell in the upper range at 594 bases. And so this indicates to us that our adaptive sampling process was currently working during the live sequencing part. And we were able to get coverage of our regions of interest. We had a mean coverage of 67.5x coverage over our 3 samples, so that is an average. And then we had about an average of 11x coverage outside of our regions of interest. So overall, we were able to improve the library preparation protocol for fragmented and challenged DNA input. We increased DNA retention, and we created a working protocol for adaptive sampling to simultaneously analyze STRs and SNPs and mitochondrial DNA. We're still working on, however, further increasing our DNA retention because it's still not ideal, and so we'll continue to modify and optimize that protocol as we move on. Again, adaptive sampling exhausts pores rapidly, and this isn't something necessarily that we can combat, except with the successive wash and reload steps to try to maintain the fuel for the nanopores that aren't collapsed. So moving forward, we'll be performing sequencing on truly challenged samples, and we'll continue the optimization of our library preparation protocol for challenged samples specifically, as well as developing sequencing parameters for maximum data output in this process. We would also like to further modify our data analysis pipeline for the streamlined analysis of adaptive sampling data, covering everything from coverage, enrichment, and variant calling. I would like to acknowledge the NIJ as well as my committee and my lab members for supporting me and funding me in this process, and I'll take any questions that you have. Yes, I think there will be a microphone.
Katherine, I have a question.
Cool beans, yeah.
So considering that most forensic laboratories or like service providers are just now beginning to sequence, but on a MySeq platform, what kind of advantages do you see the nanopore or Oxford nanopore technology providing in a forensic scenario or setting?
Yeah, good question. So ONT provides a lot of PCR-free protocols, and I know that PCR has a lot of issues with developing either primer dimers or altering variants during the actual elongation step of PCR. And so I believe that ONT might be able to mitigate some of that, as well as it's more direct sequencing rather than sequencing by synthesis. So in that case, you're looking more for the variants within the DNA and not variants that are possibly created by erroneous additions of nucleotides during the sequencing by synthesis process.
Okay, I have a question regarding the two different kits where you saw so much loss versus the other kit. I've done a little bit of nanopore sequencing, but I thought there was an optimal, so not only do you have to consider the input quantity that's recommended, but isn't there something to do with the length? So there's a compromise between quantity and length to determine the optimal going into the reaction. I'm just wondering if those differed between the two kits, like one you had 100 nanograms, the other one was 2.
Yes.
Were they the same DNA dilutions or sheared, and how that might impact?
Yeah, so in this case, our data was preliminary, and so again, that wasn't necessarily an apples-to-apples comparison, and that's totally a fair limitation. The difference between the kits in terms of the femtomoles that you would put in are slightly different, and Nanopore does really recommend their input recommendations in femtomoles rather based on mass of the DNA, which could all be altered with fragment length, rather than like nanograms or anything like concentration-wise. But the major difference between the two kits with the 96 barcodes and the 24 barcodes and why we made that switch was there's, funny enough, a typo in the 96 kit protocol where you only moved forward 0.75 microliters. And in discussion with ONT, there was a little bit of, is it a typo, is it not a typo? And so we moved forward with the 24 barcode kit instead, which does not contain that typo in the protocol itself. And that's also part of the reason why the DNA retention went up so much.
Thank you so much for the presentation. So I know that you're still in the phase of the optimization, but based on the literature that you've done, what do you think would be and probably it wouldn't be a concentration based on your answer right now, but what would be in a concentration context, the minimum input of DNA required to be able to get enough coverage for STRs, SNPs, and Mito?
So based on what we have right now, our recommendation would be anywhere in the 250 nanogram in terms of absolute mass for input. Of course, your concentration can vary by volume, but if you have 250 nanograms, it can be vacufuged, or you can elute down into what you need for the input for ONT.
So there is a concern of the error rate of the nanopore device. Would you care to comment on that, and would that be a limitation for its use in forensics?
Yeah, so ONT has been historically considered an error-prone sequencing platform. However, in 2021, they were able to release their newest sequencing chemistry, which is capable of base calling a single molecule at a Q20 or above. And so it's no longer really considered an error-prone platform. It's pretty high accuracy in terms of comparison with other platforms.
So just out of curiosity, what was your incubation time before, and then what did you increase that to?
Yeah, so some of the incubation times were like about a 10-minute difference. So really for the Ampure bead cleanups, it would be like from 10 minutes to a 20-minute incubation. But for the, did you have a specific reaction?
Sorry, for the adapter ligation?
Oh, for the adapter ligation, let me remember off the top of my head. I believe we went from, you leave it for about 10 to 20 minutes, and you upped it to like an hour.
Okay.
Yeah.
And then just one more question.
Mm-hmm.
How did you choose the SNPs that you wanted to target for the sequencing?
That is a good question, and I don't remember off the top of my head. They're SNPs that we've used in the past,
okay.
And so we just moved forward with that.
Okay.
Any more questions?